The protocol demonstrates how to efficiently generate human-induced pluripotent stem cells from 4 milliliters of peripheral blood. On day 0, isolate and expand the mononuclear cells from the peripheral blood sample. Transduce the primary cells with the reprogramming STEMCCA vector on day 9. Proceed to plate the transduced cells onto inactivated mouse embryonic fibroblasts and culture for about 3 weeks. Pick the HESC-like colonies for expansion and characterization. Results of immunofluorescence staining for the expression of pluripotency markers like SSEA4, TRA160, and TRA181 demonstrate the efficient generation of induced human pluripotent stem cells.
The use of the STEMCCA lentiviral vector to generate human-induced pluripotent stem cells offers several advantages over other existing methods. Perhaps most appealing is the overall simplicity of the approach, as it requires easy access to donor tissue, and by that I mean readily obtainable 4 ml of peripheral blood. The most important attributes of this method are the efficiency of reprogramming and the quality of the obtained iPSC clones. Demonstrating the procedure will be Andreia Gianotti Sommer, a postdoctoral fellow in my lab, and Sarah Rozelle, a graduate student in George Murphy's lab.
On day 0, draw 4 milliliters of peripheral blood into a vacutainer cell preparation.